My name is Samuel Flores, and I will be talking to you about bridging the computational skills gap, which is about my efforts to teach programming and other computer modeling skills to biologically oriented students. I apologize for not being able to attend. I'm running our graduate school's annual meeting in Gothenburg. So I feel I should begin by pointing out that bioinformatics is an interdisciplinary field. So it's by its nature, it's going to have a mixed intake of biological and computational students. And this mix is essential to bioinformatics. We cannot take only computationally oriented students, or else we'll just be answering all the wrong questions. So our challenge is to teach computer science to biologists and biology to computer scientists. But the former is the challenge that I'm focusing on in my work. Now, the approaches that we are taking to confront these challenges is first, we feel that we need to scale the ambition of the assignments according to their present skill level. We have tried to form project teams with mixed skills and experimented with the idea of rewarding students for helping each other in an equal exchange, and of course providing computational and biological remedial training. So the first and third work, and we've tried them. The middle is not so easy to implement in practice because we can't grade them on how much they help each other. We grade them on the attainment of learning outcomes. We have fertile ground for implementation. First of all, I just completed a summer course on synthetic biotechnology. And so this is an undergraduate / master's course on basic biological wet lab and computational techniques for engineering bacteria. And that's what I'm going to be mostly talking about. I also ran a course called Bioinformatics KB3004. This is a spring course for MS students plus some undergraduates. I'm going to be talking less about that because it's a different issue. I run the National Graduate School in Medical Bioinformatics, and so this provides courses for PhD students all over Sweden. And in the future, we may have an MS in bioinformatics as well as one semester bioinformatics programs for foreign students. The course was to teach the students to gain genetic control over a bacterial cell. And the specific challenge was to increase the production of color proteins. To this end, they learned computer modeling, which they then used to design the experiment, or in some cases to explain their results after the fact. Then they would order the DNA and test their strategy in the wet lab. They throughout attended lectures in the flipped classroom format. The readings were encouraged by pre-lecture quizzes. We had many evaluation elements. So like I say, multiple quizzes. Then there were 2 partial exams as opposed to 1 final exam. We graded their lab notebooks and their computer exercises, as well as their group oral presentation and their final written report. Mostly a biological intake of mostly master's students, except for one undergraduate who came from China to take the course. The other foreign students applied but had admissions trouble, which hopefully we'll be able to work on. 10 students completed the course, all of whom are passing. The final grade is not known yet. The lectures, especially the flipped ones, in my perception, moved faster than I would have expected. The projects were quite good. In 2 cases, they were completely unexpected. One might even be eventually publishable. The students perceived their own computational skills as having improved, which was the main objective. And they seemed to be, in general, quite happy. So that was good to hear. In conclusion, I think diversifying the evaluation elements was a good idea. And the Canvas learning management system was crucial for making this efficient. The flipped classroom model worked well for me. The lectures that I gave in this format were faster, and the students still did quite well on the quizzes. In fact, did even better. Scaling the challenges in the computer lab seemed to work. There were some comments in the evaluation that indicated that, and that was my impression. In terms of remedial training, what I did was spend more time with the slower students in the computer lab, and in any case, the faster students were able to finish early and start on the final project. As for next steps, I'm considering writing a pedagogical paper on the synthetic biology course, and of course a conference presentation. We're going to be moving more lectures to flipped classroom formats and recording some videos to support those. I will possibly create a distance learning course which includes a theory and the computer labs from synthetic biology, but not the wet labs. And this would run in parallel with the full residential course, which would still include the wet labs. For my bioinformatics course, KB3004, I think I will be creating Jupyter Notebook exercises. And these allow people to learn the algorithms, but would not require fully developed programming skills. And I'll, of course, incorporate more flipped classroom lectures. And here also I'm thinking about reforming it as a distance learning course in parallel with the residential course. Thank you all for listening, and if it is technically possible, I would like to take questions now.